Good morning, everyone. Today we're going to continue studying respiration, and the first thing we're going to do is a recap of what we learned from last time under the form of a equation. So the equation for cellular respiration is the following. We can deduce this from the experimental setups we had a look at on yeast in the last video. The equation goes as such. You remember that when glucose was injected, then certain gas exchanges occurred. We saw that O2 was decreasing in the medium. That means it's been used up by the yeast. So glucose and dioxygen will be transformed into carbon dioxide, water, and energy. You can pause the video and try and balance this equation. And if we balance it, it will be as such. We need to have 6 dioxygens, 6 CO2s, we'll have 6 carbons there, and 6 H2O. Under the word energy, what we mean is formation of molecules which have energy, and it can, for example, be ATP. So here we are, this is the cellular respiration equation. In the previous video, we saw that cellular respiration is actually two things. The first step is glycolysis. This starts with glucose. Glucose will enter the cell, and a process called glycolysis, which we'll study in a few minutes, will transform glucose into pyruvate. And we then saw that pyruvate can either be used in fermentation when there is no dioxygen, or it can be used by the mitochondrion. That's when there is dioxygen in the medium, and we'll see that this pyruvate will go through a cycle. You can see TCA cycle. It can also be called the Krebs cycle. So what we're going to focus on now is glycolysis. So how you go from glucose all the way to pyruvate. Number 2, detail of glycolysis. I'm just going to pinpoint a few things from the paragraph that you have in your lesson. Glycolysis, so we've got two ideas. We've got the word glucose and lysis, which means to break down. So we're going to break down a bigger molecule into a smaller molecule. This smaller molecule, as we said before, is pyruvate. And this is the formula of pyruvate, CH3COCOO-, and you have an extra proton. Second thing, the process is going to release energy under two forms. We've got two molecules. We have ATP, you know this one quite well now. We saw previously in the videos on photosynthesis that ATP is commonly produced and used in plants. And we've got NADH. You might be thinking, oh, this looks a bit like NADPH. Well, you're right in the sense that it will have a very similar function to that of NADPH. So you're probably getting this, that we've got similar things going on, just like in the chloroplasts of plants, but here we are talking about glycolysis for the moment, and we've got ATP and NADH which will be produced. And the third thing Glycolysis is a sequence of 10 enzyme catalyzed reactions. We will have a look at these different enzymes, these different reactions, and I will pinpoint what you need to know for the baccalaureate. Are you ready to have a look at this glycolysis diagram? Don't be scared. There are quite a few things on it, but you only need to know a few things. You need to understand how it works, but you don't need to learn it by heart as such. This is the diagram of glycolysis. It's a simplified version because if you remember in the text we said that there were 10 enzymes. In this one they are not all figured, but you've got the main steps of glycolysis. Let's explain some of the symbols. C6, C3, this means that the molecule has 6 carbons. We can see that in the first steps we always have a molecule which has 6 carbons, and then at this point here, this molecule with 6 carbons will split into 2 with 3 carbons. What else? We also have phosphate groups. You can possibly see the P symbol, so we've got a phosphate here. We've got 2 there, 1 here, etc. In the process of adding or taking phosphates, this will imply that either ATP is used up or ATP is produced. So to keep track of is there going to be more ATP at the end or less ATP at the end, we are going to have a ATP count, and we're also going to check how much NADH2 is produced or taken in. So we'll also have an NADH2 count. We're going to go step by step and count the ATPs and briefly explain what's happening from each step. Here we go. We start off with glucose. This glucose, so the arrow here, there really should be an E for enzyme, because between each molecule there's actually the intervention of an enzyme. You remember, an enzyme is a protein which will speed up a reaction. In this case, this enzyme would speed up the reaction of adding a phosphate group onto this molecule. So when we go from glucose to this first molecule, which has a phosphate group, we are using the phosphate group from the ATP, so that means that 1 ATP is used up, so I'm going to put -1. In the first step, 1 phosphate is added, so that means 1 ATP is used up. Second step, not many changes here. Third step, again, we're adding a phosphate group to the molecule, so 1 ATP is being used up. Oh, I've just realized that the previous one was erased, so we've got -1, -1, so that's -2 so far. Moving on. In this step here, we have the production of 2 NADH2. So that's something we can add here in the NADH2 count. Then 2 ATP will be produced. Indeed, we're losing the phosphate group for 2 molecules, so 2 phosphates will be released, and that can regenerate ADP into ATP. So we have +2 here, and if we continue, last step, we've got the regeneration of 2 ADP into 2 ATPs, so again +2. If we do a sum up, In glycolysis, we go from glucose to 2 pyruvates, and in the process, -1 plus 2, we can cross these ones out. We are left with +2. That means that in this big process of 10 reactions, 2 ATP on the whole will be produced, as well as 2 NADH2. We saw it here. 2 NAD will be transformed into 2 NADH2. I said transformed, we could be more precise. We can say that they have been reduced, as it's a gain of protons, gain of electrons. There we are. We are now going to do a recap in a smaller diagram, just for you to know what are the important things. Task: make a diagram showing the overall chemical reaction of glycolysis. The reason we're doing this is because your syllabus states that you need to know the names of the substrates and the final products of glycolysis. And I think that this type of diagram is the best way to do it. And you'll also know what's happening during the glycolysis, and that's what you need to know. As we said, glycolysis starts off with this molecule here, C6H12O6, which is glucose. This glucose is going to go through different reactions, 10 reactions, 10 enzymes transforming different molecules. At the end, we end up with CH3COCOOH. This is pyruvate. We can see that glucose has 6 carbons. Pyruvate has 1, 2, 3. Therefore, it will be 2 pyruvates in order for the equation to be balanced. In the process, remember the ATP count and the NADH2 count. In the process, we've got 2 ADP plus phosphates, which will be regenerated into 2 ATP. And secondly, 2 NAD will be regenerated into 2 NADH2. NAD being reduced, there's a gain of protons and a gain of electrons. This you can learn by heart, as you need to know the names, glucose, pyruvates, and the formulas. You need to know C6H12O6, and you also need to know CH3COCOOH, and you need to know what's happening overall during glycolysis, and you now know that 2 ATP are produced and 2 NADH2 are also produced. So here we are. This is the general recap of glycolysis. We've covered the different steps of glycolysis. What I would like to do now is just show you where different things take part in the cell. What you can do is take your booklet, page 12, and we're going to start talking a little bit about this diagram here, which is a bit of a general recap. The first thing I would like to do, and what you can do at the same time, is grab a pen and show a very important limit, which is this one here. You can read CYT. I think they've done a typing error. It says cis, but it should be CYT really. This line here is the limit between the cytosol and the mitochondrion. So the cytosol will be this top part, and this bottom part will be in the mitochondrion. So the first two labels, CYT for cytosol and MIT for mitochondrion. What we can see is that glycolysis and fermentation will take part in cytosol, whereas the Krebs cycle and oxidative phosphorylation will take place in the mitochondrion. I'm just going to give you the names I've just said so that you have them. G is for glycolysis. You can recognize the different steps. You can see that we start off with glucose here and we end up with 2-pyruvate here. So the pyruvate can either be used in fermentation or it can be used in the Krebs cycle. There are two different types of fermentations. This will depend on different organisms. We'll have either one type or both types. FL is for lactic fermentation and FA is for alcoholic fermentation. We will study these in detail and we'll see that they have many uses, especially in the food industry. Krebs cycle, that's the aim of the next video. In the next video we will detail the Krebs cycle, and along with that we will have a look at oxidative phosphorylation. You can probably see this diagram here, and it maybe reminds you a bit of the Z scheme in a way, and we'll see that there are similarities with, we'll talk about redox potentials and different proteins which will transfer electrons. And we'll see that this is in a sense similar to what we saw in the Z scheme. So here we are, that's all for today on glycolysis. And to finish the video, I found a new version in a way of the cup song. You might have heard of the cup song, and this is a glycolysis version of the cup song. I'll let you listen to that. Thanks for watching and see you in the next video. Bye.